Today we're on lecture 17. This is going to look at immunization. So immunization is the administration of antigen inoculum, and these are called vaccines. With active immunization, it's administered to stimulate an immune response in the patient, where with passive immunization, the patient's going to acquire temporary immunity through the transfer of antibodies from an immune individual. When we're looking at a titer, this is the antibody level in the blood. It can be a measure of vaccine effectiveness. Just a brief history, smallpox was one of the diseases that was helpful in coming up with vaccination. It was noted that children who recovered from smallpox did not get it a second time. So people deliberately infected young children with the ground smallpox scabs, and this reduced the mortality and morbidity. Edward Jenner demonstrated protection from smallpox with inoculation with crust from a person who was infected with cowpox. Cowpox just happened to be similar enough to smallpox that it would give some immunity. Another person who made significant contributions here was Louis Pasteur. He conducted experiments with Pasteurella multaceta, and he demonstrated an effective vaccine against the organism. In the late 1900s, we had formulated vaccines for polio, measles, mumps, and rubella in the U.S. We now have vaccines for chickenpox, hepatitis A, hepatitis B, and many others. So there's different types of immunization. With vaccine types, not all are equally safe or effective. A live attenuated is a modified live vaccine. These tend to elicit vigorous responses, and they may provide contact immunity to those around the person that was vaccinated. They reduce the virulence so that they're still living, but they're no longer causing disease. These can be grown in tissue culture until they lose their virulence or are genetically manipulated. These are a risk to give to pregnant women or those who are immunosuppressed. Inactivated vaccines are going to be formaldehyde inactivated. Well, whole agent vaccine is the deactivated whole entire microbe, where a subunit vaccine is just going to have antigenic fragments of the microbes. So recombinant vaccines can be produced by genetic modification. They're safer, but they cannot replicate, so you need boosters. Adjuvants are substances that will increase the effective antigenicity of the vaccine. They may stimulate inflammation and allergy. Killed vaccines are recognized as exogenous, and they will give a humoral response. The VLPs, or virus-like particle vaccines, resemble intact viruses, but they don't actually contain any viral genetic material. Toxoid vaccines, these are chemically or thermally modified toxins in vaccines that will stimulate immunity. This would include things like tetanus and diphtheria vaccines that will stimulate antibody-mediated immunity. They've got few antigenic determinants, so you do need multiple doses and boosters of this. Conjugated vaccines were developed to deal with the children's immune system, but does not deal well with vaccines that are based on capsular polysaccharides before they're 24 months old. What they found was when the polysaccharides were combined with proteins like diphtheria or tetanus toxoid, they were better tolerated. So now those vaccines are given as a combination. Nucleic acid or DNA vaccines, these are plasmids of naked DNA that are injected, and this results in production of the protein antigen that's encoded in the DNA. So when we look at technology in vaccines, they're using recombinant DNA a lot. You can detect virulent genes and produce pure bacterial and viral antigen, but they're less effective. The genetically engineered microbes can be used as vehicles. So in vaccine manufacture, bacteria can be grown in mass in culture vessels, but viruses need to be cultured inside sterile chicken eggs. So that means these cannot be given to those with egg allergies. There are vaccine schedules that are published as recommendations for adults, for children, and for special populations like those who are HIV positive or healthcare workers. So when we look at active immunization and vaccine safety, it's going to have mild toxicity. There's usually pain at the injection site. Poking the skin with a needle is not going to feel good, even if it only has saline in it. and can lead to malaise and fever. Anaphylactic shock is when a person has an allergy to a component of the vaccine. Some vaccines will have residual virulence that can actually cause disease. There have been several lawsuits. The Vaccine Adverse Event Reporting System is used to keep track of adverse reactions that occur with vaccines. So when we look at passive immunization or immunotherapy, this is the administration of antibodies to a patient. An antiserum is going to contain antibodies for passive immunization. It's typically collected from large animals that are intentionally exposed to the disease. There are some limitations to the antisera. It can contain antibodies against many antigens, not just the antigen of interest. And repeated injection can actually trigger an allergic response or serum sickness. It may contain viral pathogens, and the antibody is degraded quickly. Its half-life is only about 3 weeks. Antitoxins are antibodies that are directed against a toxin. Antivenoms are going to fall into this category. So again, with antibodies, they're very specific. If you are bit by a snake, it is important to know what kind of snake it is, or at least for you to be able to recognize the snake, and hopefully someone will be able to say from your description of the snake what kind it is to give the appropriate antivenom. So if you give an antivenom to a different kind of snake, it's not going to be effective, and there is always risks with antitoxins and antivenoms. Hybridomas are going to be tumor cells that get fused with antibody-secreting plasma cells to produce clones that will secrete large amounts of a single antibody. This will help with some of the limitations of antiserum. With monoclonal antibodies, an identical antibody is secreted by the clones originating from a single plasma cell. We're also working on chimeric monoclonal antibody. Here they use genetically modified mice to make a human murine hybrid. These will make an antibody that's 66% human. This is used to make rituximab, which is used to treat some leukemias and other autoimmune disorders that are going to have excessive B cells. With humanized antibodies, these are constructed so that the immune portion is limited to the antigen binding sites. The rest, 90%, is human. This will lessen the response against them. An example is trastuzumab or Herceptin, which is used to treat breast cancers. So herd immunity, this provides protections to all individuals due to the inability of the pathogen to effectively spread in that population. This occurs when most of the population, it usually takes at least 75% being resistant. So with immunotesting, we can use known antibodies to detect antigens, or use selected antigens to detect antibodies in a patient's blood. Serology is just the study and diagnostic use of antigen and antibody interactions in the blood or serum. So with precipitate tests, antigens and antibodies are mixed, and they'll form a lattice-like precipitative immune complexes. It is important that you mix these in appropriate proportions, or you'll have excesses of one left over and it won't look like a precipitate. With immunodiffusion, you create two wells in an agar gel. The wells of the antibody and the antigen are allowed to diffuse, and where they will meet, a precipitate forms. With agglutination tests, you can have direct agglutination tests where the antibody can cross-link a particular antigen. The agglutination involves the clumping of soluble molecules. It's easy to see and interpret without visual assistance, so you can do serial dilutions to determine the titer of an antibody with agglutination activity. With seroconversion, this looks for a rise in the titer. The titer is higher later in the course of a disease than it is at the outset. So you can look at the concentration of the antibodies and get an idea, is it a new disease or is it just one that is reactivated? With indirect agglutination tests, the antibody reacts with a soluble antigen that's been absorbed onto particles such as bentonite clay or latex beads. So here you watch for the particles to agglutinate, and you can use the particles also coated with an antibody to detect the antigen. So using the particles just makes it a little bit easier to visualize. With immunoelectrophoresis, gel electrophoresis is used to speed up the movement of the antigen and the antibody in the gel. Again, you're looking for that line to precipitate. In neutralization tests, the antibody can neutralize the biological activity of many pathogens and their toxins. So if we look at viral neutralization, viruses are mixed with a specific antibody, and when the antibody is going to attach to it, they will lack their cytopathic effects. So when it's introduced into a cell culture, you're watching for it to prevent the cytopathic effects in the culture that the antibody neutralized the virus. With viral hemagglutination, this can be used because some viruses are not cytopathic but will clump red blood cells. So they'll produce viral hemagglutination. With complement fixation tests, this will detect the presence of antibody in serum too small to detect with agglutination. This isn't used a whole lot anymore. It's pretty much been replaced with ELISAs or a genetic analysis now. So turbidimetry and nephelometry, these are automated methods that will measure the cloudiness of a solution when the antibody and antigen are mixed together. So when they clump together, it's going to make a more cloudy solution. With labeled antibody tests, the antibody is linked to a molecular label that will allow them to be more easily detected. Direct fluorescent antibody tests are ones that will identify the presence of an antibody in a tissue. You flood the tissue with a radiolabeled antibody and then wash it to remove the unbound antibody. If it sticks, your antigen is present. With indirect fluorescent antibody tests, you identify the presence of antigen in cells or tissues or in serum. The antigen gets fixed to a slide, and then the patient's serum is added, and then a radiolabeled anti-antibody is added. So ELISAs are enzyme-linked immunosorbent assays. The label is not a dye, but an enzyme that will react with a substrate to produce a colored product. And this is what will indicate a positive test. So you start out with wells in a plate that are coated with antigen. The excess antigen is washed off, and another protein, gelatin, is added to coat the polystyrene surface that's not coated with antigen. The serum sample is added, and if the antibody is present, it will bind the antigen. The anti-antibody is labeled with an enzyme that's added, and then the enzyme substrate is added, and the reaction will produce a visible color change. So immunoblots or Western blots, these will detect antibody against multiple antigens in a complex mixture. These are used to verify HIV tests in ELISAs. It will detect more types of antibody, and there's less misinterpretation. So the antigen in a solution is separated by electrophoresis, and a protein band pattern is then blotted onto a nitrocellulose membrane. It gets cut into strips, and then ELISA is done on the strips. It's washed and exposed to serum, and then you wash and expose it to an anti-antibody, and then wash and expose to the substrate. So point-of-care testing, this allows healthcare providers to test directly without having to send things for the lab. There are really big advantages to having rapid results with these. So the immunofiltration assays, these are rapid ELISAs that use antibody bound to a membrane filter instead of a polystyrene plate. The larger surface area is going to reduce the amount of time it takes. With the immunochromatographic assays, these are faster and easier to read immunoassays. The antigen solution, such as the infected blood, flows through a porous strip and then encounters the antibody labeled with pink colloidal gold or blue colloidal selenium. Where the antigen and antibody complex binds, the colored immune complexes form as a pink or a blue line. So this is what's done with pregnancy tests. It's looking for HCG in the urine. These can also be done for HIV, RSV, E. coli 0157H7, and group A strep.